The main functions of the electronic system are to convert light signals into numerical data, eliminate small signal events such as noise and debris through threshold, and attribute signals from multiple lasers to the correct cell or particle. How does this happen? To convert light into data, 3 components are needed: photodetectors, amplifiers, and signal processors. These electronic components work in a sequence to convert light into current, current into voltage, and voltage into data that can be further analyzed. Click each electronic component to learn more. Photodetectors are light sensors that can detect photons of light. Incoming photons cause photodetectors to produce electrical current. Amplifiers convert electrical current from photodetectors into a voltage. The resulting voltages are larger in magnitude than the incoming currents. Signal processors quantify voltage pulses, providing numerical values for the pulse height, width, and area. Data is received and stored by the host computer. The computer enables statistical analysis and graphical display of data. The computer workstation stores and displays data for further analysis in the form of CSV and FCS files. Recall that photodetectors are used to detect light in the form of photons and generate electrical current in the form of electrons. Photodetectors vary in quantum efficiency, which is the number of electrons they can generate per incoming photon. Click each topic to learn more about photodetectors. Among the photodetectors used in BD cytometers, photodiodes have the lowest efficiency. They generate only 1 electron per incoming photon. Typically, photodiodes are used to detect very strong signals, such as forward scatter light, FSC. Photomultiplier tubes are highly efficient detectors, generating millions of electrons per incoming photon. PMTs are used to detect weaker signals, such as side scatter light and fluorescent light. Some cytometers use PMTs for FSC detection of small particles. What makes a photomultiplier tube so efficient? Click the next topic to find out. Photomultipliers consist of a vacuum tube with a cathode, focusing electrodes, dinodes, and an anode. When a photon of light enters a PMT, an electron is generated at the cathode. Focusing electrodes guide and accelerate the electron to a series of dinodes. Each time an electron strikes a dinode, additional electrons are generated. With this cascade effect, millions of electrons can be produced from a single incoming photon. The voltage applied to each photodetector can be adjusted through the cytometer software. What effect do changes in PMT voltages have? The PMT voltage affects the display of the data. See how populations shift along the side scatter axis when its PMT voltage is increased or decreased. Notice that the data spread along the forward scatter axis remains unchanged. Recall that voltage pulses are generated each time a cell or particle passes through a laser beam. Through signal processing, the voltage pulses are digitized and converted into numerical data. Accurate signal processing requires laser delay adjustments in cytometers with more than one laser. Thresholds determine whether signal processing will occur. Click each topic to learn more. Laser time delay adjustments are made to ensure that signals from each laser are attributed to the correct particle or cell. In cytometers with multiple lasers, the laser beams are spatially separated so that a particle passes through only one laser at a time. The spatial separation of the laser beams results in a single particle generating signals at different moments in time. Laser time delay electronics ensure that all parameters for each particle are processed together as one event. In this example of a 3-laser system, a particle passes the red laser first, then the blue, and finally the violet. The laser time delays are adjusted to ensure that the signals are attributed to the correct event. Pulses are generated whenever particles pass through the laser. To avoid the collection of data on debris, a threshold can be applied. The threshold is the minimum pulse height for signals to be processed. When a pulse exceeds the threshold, signal processing on the pulse occurs. Signals below the threshold are not processed. No data is recorded for those events. In this example, a threshold is set on the forward scatter parameter to eliminate small signal events, such as noise and debris. Notice the differences in data display for the 2 forward scatter thresholds. Signal processing results in pulse quantification. Numerical values are generated for pulse area, height, and width. This data is then transferred to the cytometer workstation for storage and analysis. Reflect on what you have learned in this module. How does the electronic system convert light into data? What is the purpose of the laser time delay electronics? What are the functions and applications of threshold? Click Next. Now that you have completed this module, you should be able to describe how the electronic system converts light signals into numerical data. Explain how laser time delays ensure that signals from multiple lasers are attributed to the correct event. Explain how and why thresholds are used.